Like a little alien creature.
Mm-hmm. Going ahead.
So one of the reasons we're particularly looking out for sponges is last time we collected sponges in this area of 12 that I was able to look at with an expert sponge taxonomist. 4 of them turned out to be new species.
Ah, it's amazing.
Oh, wow.
Yeah.
Yeah, with all of the corals and sponges from these depths, you're finding new things all the time, particularly with the sponges and even with the corals. Even some of the groups that we think we know really well, once we start collecting more of them and looking at them genetically, it is not uncommon for new species to appear. And a lot of the species descriptions that we do have were written in the late 1800s and have just been used since then. So now that we have more modern techniques, genetics, and scanning electron microscopy, we're getting a better picture of the species that actually live down here.
Yeah.
Shot right now, we have 2 thorny heads, a big one and then a tiny one. Like it's pretty young. Just went off the right-hand side of the screen.
So it's still quite
when they
deep.
first land, they'll actually be.
We'll see more rockfish species. Sorry, Meredith.
Oh, yeah. Yeah, most of the rockfish, while they can be found down this deep, it's very rare. That usually start about 600 meters. Where they start getting more, 500 or 600 meters is where they start getting more of them. Thorny heads are found throughout. The poor thorny heads have a very unfortunate nickname. They get called the idiot fish. And there's been a long discussion as to where that name came from, whether it's about something as the fish themselves or whether it comes from the fishing community, because if you pick them up, their whole head is just this mass of spines. And so it might be more about your level of intelligence were you to grab them by the head.
Anemones and sea cucumbers, the solus again.
Yeah, I don't know about the earlier part of this dive, but I haven't spotted any bubble plumes or microbial mats on this.
Yeah, there hasn't been really any sign of like even really reduced sediment, but we're keeping our eyes out and we will flag it if we see it.
Yeah.
Eel pouts. Might be nice to do a zoom on one of the eel pouts, Meredith, for Tom if we come across one. Just to confirm.
Yeah, sure thing.
Thank you.
Or Paracetamol. That's
So
these
these
little
video
white.
transects.
Go ahead, Abby.
Oh, it's going to just,
These little
all
white
the
dots.
video transects are going to be analysed by the Southwest Fisheries Science Centre. They have a protocol that they've been using for a number of years now on ROV footage.
Another one of these corals that's been overgrown by zoanthids, and then another bubblegum.
Forest of them again.
Oh, nice. Now let's explore through here.
Oh, that's gorgeous.
Yeah.
Nice.
Wow.
Crab too.
Oh, wow.
Right on the side of a cliff. Beautiful.
Oh, with some nice sponges there too.
Yeah,
we get a shot of that.
So is the still camera taking photos at the same time as the screen grabs?
I'm not sure.
Either of them will work.
So.
That is beautiful.
Yeah, you want to take a zoom on this guy?
Yes, please.
Yes, please.
Oh, it's sponge.
That's tricky. Which guy?
Gonna come a little wide.
So you often get.
Is it a little wide? You want to come all the way?
No, this is all right.
So you'll often get large sponges on the sides of cliffs like this because the currents tend to be higher, which brings them food, sort of facilitates their filter feeding.
All right, you want to come full wide, and then I'm going to go over here and see what there is to see. It's a really nice community.
Mm-hmm.
I'm just holding off on ship moves so you get a chance to take a look. Sure thing.
You can see in the upper one there's a nice crab hosting in that sponge.
Oh yeah. You want to go ahead and do a partial there, Al? When I'm back. That's good. All right.
So the morphology of this one is quite different. See, there's ridges on the inside rather than.
Mm-hmm.
Yeah, ridges rather than no holes.
Are you going to come wide there, Al?
I got a barrel sponge right there underneath that header coney, the lower left of the bubble gum.
All right, Al. Come push in.
Also a really nice view here of like what that rock morphology looks like, which is almost like a sedimentary, but I'm not a geologist, so.
Lots of sediment-free substrate though that sponge larvae would be able to attach to.
All right.
Sediment substrate.
I'll go ahead and come full wide there, Al. Sorry?
So I was just looking at the base, trying to work out if that's just the base of the heterocone or if there's a different. I think it is. I think it's just the base of the heterocone. There's another glass sponge called Tritodictyum, which we're on the lookout for, but I don't think it's that.
All right, are you guys satisfied with this area? Should we keep pushing on?
Can we get just one zoom on that coral that just went off the top of the screen, just above the lasers?
Sure thing.
Let's see what it is.
Is it a bubblegum coral or is it?
I think it's another primnoid. I think it's another peristinella.
Okay.
It's just below the bubblegum that's coming into the screen.
Oh, I see. So it's
lighter pink one.
Copy. I'll drop down a little bit and we'll get some light on it. All right, Elle, you want to go ahead and push it on this guy? Stand by.
That's great.
Let me get you a better shot, or is that all right? Sorry, science.
That's all right. That's great. Thanks. Yep, it's another peristinella.
All right. Go ahead and come light there, Al. Should have stopped yet. Argus is hanging out just west of your field of view. Yeah. All right, science. Are we all good with that spot? Boogie onwards?
Yeah. Yep, we can keep moving on.
Just gonna wait till you're kind of back solidly in the field of view. Sure thing. Yeah, it looks like I'm about 10 meters out. Yep. I think it's gonna start trending like 070, but I'm nervous to the last one to trying to decide which is more trustworthy. Contours are actually a better indication last time.
Nice snail trail through the wall of mud there. Well, I'm always impressed with how some of these invertebrates, especially the ones like the snails that seem a little heavier, can just cling to these steep, muddy faces. Got a C-pen there, just one above the laser, little guy.
Back into mud.
Yep. Just get these little patches of rocks that are these little islands of biodiversity, and then back out into the mud. Just when you think you're just in mud, you get more sponges. You get more of these ledges off this space. We're in this canyon, and we're kind of following the depth contours, slowly getting shallower.
Ridge nav, another step, same bearing.
Following the ridges. And the map we have of the bottom here does not quite show all of the topography. So as the ROVs are exploring, they're having to kind of maneuver around these micro features that just don't appear on the topo bathymetric map we have at the bottom. It's those kind of features that can provide some great habitat. Thorny head there.
Meredith, I know that different morphologies of the heteroconies are on our collections list. We've seen quite a few of them. So if we do see a smaller individual, I'd also be keen to maybe try and get a stick.
Yep, well, it looks like we're back into some of them. So if there is one that looks like a good size for a piece of, just,
yeah.
Sure thing. And,
and
oh, sorry. What sample are you talking about? Are you looking for a sponge like this?
Yes, we're looking for a sponge like this
Okay.
that is in a good position for the pilots, and then just a snip. We don't need to take a big piece.
Great. You gonna stop the ship? Sure. Bridge and nav, hold position. There's only a 10-meter move, right? That was baked in. Let's see if I can.
They're pretty fragile. They will tear quite easily.
Okay.
They have these little
So are
projections
you
on
full
the
wide?
outside.
Do you guys want to slurper this guy?
do you think adding slurp
Like
or?
I think it's soft enough that it would, yeah, that a piece will just tear off in the slurp.
Sure thing. That's great. Thank you. All right. Let me see if I'm.
So did you want to try and sample it or not?
Yeah, we're getting set up right now just to
Okay.
make sure we're stable.
Sure.
Try that out. Oop. Okay. Sounds good to me. Oof.
So we're going to be taking a slurp sample?
Yeah.
So the next open one is number 3.
Oh, maybe we should verify. I was assuming that we could take a slurp because it was friable. Science, can you verify that you want, what type of sample you want, slurp or snip?
Abby?
I
Try with
think
the
I
slurp.
think it's very fragile, so I think a slurp will be good.
A slurp? Sorry.
Yeah, I think so.
Yeah.
Okay, great. Would you mind getting up the bucket view there, please? And we'll go ahead and flush.
This is one of the glass sponges, and coastwide we have several different morphologies of these. And Abby is interested in knowing how many species there actually are. So we're sampling to do taxonomy.
Science, are all the slurp jars open?
So yeah,
Except for number 1.
the next available one is number 3 actually.
Number 3,
Okay.
Roger. I didn't see a vortex in there. Should we try flushing again? We should go back one more, because the flush is in the back there. Oh, now we're on 6, so I think it has to go the other way. Yeah, sometimes it gets a little stuck. There we go. Yeah, that one's it. Seeing any flow? I'm not seeing flow going through there, hey? Huh. Try cycling power to it again. Wonder if it's not lined up. Yeah. Let me try that. Maybe it wasn't lined up all the way. Good. I can't tell if it's just the particles in the environment or not. Perhaps we should try one of the chambers that's open, and then try flushing into that if it lines up properly, and if not, then you might not be able to use the slurp. Science, was number 1 collected in the last dive, or was that kind of a sample that fell in?
I believe they tried to. No, they were trying to collect a sponge.
Sorry?
They were trying to collect a sponge, and it was much too robust to sample with the slurp, but we think it probably took a bunch of the spicules off that sponge that may have ended up in the first in slurp 1. So just to avoid cross-contamination.
Okay, so they did notice that they had flow though. They were able to get some sample into the chamber?
I think they.
I mean, it looked like bits of the sponge are coming off. Yeah.
Okay. Okay. Well, then that's interesting. Yeah. Yeah, let's try that out. That's a good point. I'm bouncing around a lot more now. You're on 80%? Okay. Roger. Let's just see if we get anything here. I'm not noticing anything going in there. All right.
Okay.
I'm gonna reposition myself a little bit and then we'll get that sample.
Thank you.
Sure.
Yep, sounds great. Thanks.
Yeah, I think that's a great idea. All right. Yeah. All right. Anyone want to press stick lock for me? Yeah, go ahead. Try again. Okay, you want to go ahead and try that again? Aaron? Yeah.
Yeah, so to clarify, this should go to slurp jar number 3.
Sure thing. Let's see. Not very happy with that. All right, well, give it a shot. Would you mind lining it up on number 3? Yeah, that's great.
Right.
All right, science. Do you want to zoom on this guy before we take a slurp?
Yes, please.
All right. All right, I'll go ahead and do a quick zoom there. One.
Do you want to go in? Yep, go.
Falling off there. All right, you want to?
Lovely, thank you.
Great. And come full wide, please.
Yep.
I'll do a quick sample, I think. Otherwise, I'm gonna get bumped off. Are you gonna do a partial zoom?
That look good?
Okay. That's sample number 26.
That's plenty. Thank you.
Okay. Okay, great.
Try
I'm just trying to get this guy to jostle in here.
Yeah.
All right. Is
So
there
that was 27?
No.
26. 26. Got it.
Do you want a little
Did it
piece
go
more
in?
since this one's not going in? Science?
Did some of it, I don't actually have the view of the jar. Did some of the tissue end up in the jar?
Yeah.
Let me see if I can put up the jar
Okay.
for you.
Yeah, let us know if you want a little more than that.
I think it's probably all right. We've got some in there.
All right.
Channel 3 has the jar in there.
Yeah, there's definitely a piece
Oh.
kind of up along
Yeah,
the...
that looks good.
It went
All right,
in.
you want to come
Okay.
full wide out, please?
Got it.
I think another little piece just went in. Brilliant, thank you.
Sure. Number 3. I'm not sure how to do that, yeah. I think that's, did you want me to ask video? This one here? Sorry?
Give me a second, I haven't done that yet. I'll have it. Yeah.
All right, science, so next objective, just continue on our track.
Yep, let's just
Sure.
move on up the track. About how far have we come since the last transect?
We've come far enough, but what I was just thinking, we just very recently crossed over the next waypoint. But what I was thinking, if you guys don't mind too terribly, if we can get around this spur, we'll have a nice, like, good straight run where we could do a transect with maybe not hurting ourselves.
Yeah, that sounds great.
That's okay? Okay.
Yep.
Yeah. Yeah, it looks like a good chunk in there.
Oh, look, another blob's open.
Oh.
Sorry.
I'm just taking a look at the map. So I think we should just try to like do the exploration of it till we get in and around here, and then we'll be able to do a nice transect up. Sound good?
Yeah, nice trace draw would be good, yeah. Oh. Change my Z bias for that, and now realize why I'm sinking. No, that sounds good, Aaron.
Okay.
How far into that waypoint then, if we go around the bend?
Let me see. It might be close to 100 meters till we get out around.
Okay.
All right, so we're gonna do a ship move 075.
rudder. All right, ready? Sounds good to me. All right, bridge nav. Can we get a 10-meter step 075? Oh yeah, that'd be great. Oh, yeah, that'd be great.
Just like that, we're back into some mud.
Mhm.
You want Zeus up there? I can do that. Oops, wrong Zeus.
Yes, and so where kind of Argus is, if you see HIPAC, is just crossing that second waypoint. Yes, and
Can we get the high pack back up on channel 3?
You got it.
Thanks. Perfect, great.
Good plans?
Oh, that's good.
Mm.
We did
What?
have someone onshore asking about whether there were any signs of seeps, and these clams
bridge nav,
might, if
another
they're
step,
destined,
same bearing.
these might be the first sign we've seen. Although it doesn't look like any are alive. Hash.
No.
You want to zoom in there, Al? Just a partial zoom. I don't know if that's helpful for them or not, but
Nice to see.
all right, go ahead and come wide.
The overall goal on this cruise is to look at both methane seep habitats along this part of the Cascadian margin, and then also to support to explore the seeps and coral habitat. And so we've got dives planned targeted towards each objective, but
Sounds
they
good.
do overlap. So while we're exploring these coral habitats, we're also keeping our eyes open for seeps, and we're marking the corals we see on the seep dives as well. We've got a nice coral right there.
Mm. Is that a fly
Another
trap
one.
only?
Yep, we've got some friends behind them.
We'll go see those in a moment, but for now, there's one here.
Mhm.
We're a little bit low on, we're running out of tether, so
Yeah.
we can look at this guy.
Oh, that's lovely.
Big bubble gums, got a Postolastra. That's a type of demo sponge. The other ones are actually more common in shallower water. Next to the little brown barrel-y sponge is a glass sponge.
Yeah, that sounds good.
Jenny has joined us from shore, and she's asking if we're getting some good centerfold shots, and I assured her that we are. That we've gotten some great zooms on some of these big bubblegums with their polyps out. And these two don't have as many of the ophiuroid stars up in their arms, but yeah, some of them have been like little Christmas trees decorated with ornaments of ophiuroid stars. And what no one can see because I'm broadcasting from home is that I'm doing little hand motions for the stars, sitting here watching the dive from my couch.
Would you mind coming down a little there, Kim?
Got a different type of glass sponge here. Also taking advantage of the currents in this outcrop of the bay.
Roger that.
The area we're exploring right now is a recently protected essential fish habitat area called Biogenic 2. And so because there's been this diversity of corals and sponges here, it's an area that's been recently closed to fishing in a very recent set of fisheries boundary changes.
What was the last move you just dialed in? Yeah, maybe like a 030 or something like this to cut it a little north.
Could we look at the big sponge just up at the top of the screen? It's just gone out, right?
Yeah, copy. Yeah. Yeah, please. Great. Once you come down, I'm going to lateral along this face.
Dead sponge there.
See, these rocks look darker than the ones we saw just 10, 15 minutes ago.
Copy.
I don't know if that's a crust or if they're more basaltic. More primnoid corals just at the top center of the screen, and then more of these really big, healthy bubble gums.
Sure.
In the frame on the right.
Yeah, that's good, yeah. Yeah.
Kind of steep terrain with this much coral makes life slightly more exciting for the pilot. They're doing an amazing job getting us these shots and maneuvering around the terrain.
How shallow can you get these big fans of Paragorgia, Meredith?
You'll get a species composition change, but the shallower species that we get above about 500 meters, I've actually seen on scuba at about 160 feet in a fjord in Canada.
Oh, wow.
But that's pretty rare. Typically, they don't start till about 100 meters, but in the fjords, they'll come up shallower than that.
So similar to some of those glass sponges.
Yeah, that's a different species than this one. This one you only find down below about 800 or 900 typically. Although we say all these rules about depths and stuff, and then as you explore more, you always find exceptions places.
Right. I remember seeing a piece of Paragorgia that had been brought up in a trawl. I again off New Zealand, which is kind of about as thick as an arm.
Mm-hmm.
It's incredible.
Yeah, some of these big fans we're seeing down here at the base, you can really see that some of them are probably our arm thickness around. This is a species that can get quite massive.
Mm.
At this size, they're quite old.
She's got some dead tissue on it, by the look of it.
Mm-hmm.
Many
Started the dive, we started the dive, we were seeing some of the Tritonia nudibranch that will feed on these, although I'm not seeing any across, which is nice for the coral.
Mm-hmm.
Although some of the bare branches we see might be the result of feeding by nudibranchs or sea stars. Those big rock fins seem to be definitely where we're seeing the most concentration of corals and sponges.
Yep.
One of the things we'll be looking at as we analyze the data from these dives is if we can kind of, we'll look at the ROV track and look at the directions that they were pointing and see what the aspect of the coral communities is in relationship to the rest of the slope. And you can learn more about their preferred habitat and then use that data for modeling to find more corals and more habitat for future dives and exploration.
Yeah, one of the things we get from this kind of study is that really fine.
Bridge and I have another step, same bearing. I think our next step should be due north. Sounds good, yeah.
Which corals are we looking out for particularly here, Meredith?
If we get into another community with some that are well positioned, I may try to have them collect one of the Peristinella. I believe that is on our list. I'm going to double check. We're also looking for a couple other bamboo species. We have already sampled one of these big Paragorgia, and we're looking for some black coral species, although the only one we've seen so far has been one that's pretty common, and so it's not on our target list right now.
Yeah.
Actually go 010 for one, see how that goes. Bridge nav, 10-meter step 010. Science, did you still want that zoom on an eel pout?
Yes, if we can. I think I see one up in the upper left.
Yeah.
There's also
Or
one center
lower
screen,
left,
is that one?
both.
Oh, yeah.
Okay.
Yep.
Not sure if that request was still valid or not. Okay, go ahead and zoom on in there.
That will make Tom happy.
Just really helps the ground truth, everything that
Oops, stand by, sorry.
they'll be seeing in the ROV transact.
Sorry about this. Gonna come full wide there for a sec, Al. I'm gonna just set us down since he's pretty small.
Okay.
All right, you wanna go ahead and push on in?
That one looks like it's just long enough to be counted on transect. Usually use a threshold of 10 centimeters.
Yeah, I reckon if its tail was out straight.
They're quite cute.
All right, you wanna come full wide there, Al? Like that. Great. Bridge nav, another step. 010.
Oh, and looks like Tom has just joined us. He just reached the deal tower.
And the sculpins. Morning, Tom. You're getting set up for another transect in a bit. It's good timing.
Yep, so Argus is still, we're still moving ship 010. Might bring it over 020 if we start creeping in on that again. Does that sound good? Okay.
That sounds great.
Shell hash.
Bridge nav, 10-meter step 020.
No tube worm cases.
Yeah, sure, we'll take a look here. Roger. All right, Al. Do your magic.
Abracadabra.
For Tom, who's watching, he's
Let me know if you want to go inside her.
asking about the train.
No, I think we're
What
good
was it
farther
being
out
used?
for now. Just gonna drift over them.
So far, Tom, you've gotten unlucky, and most of the big rocky habitat has been off transect, and not by
Go ahead and come wide
intention.
there, Al.
We're hoping to get you soon. Thorny
Morning.
head in amongst the tubing.
Good morning again, everyone. This is Andrew. I just saw your really nice seep discovery.
Mm-hmm.
This is really exciting.
Do we need to do anything here? Do you want us to hold up?
I'm not leading this dive.
Or if you want us to pause and sample clams at any point, let us know.
Yeah, like these right in front of us. Bridge hold position.
It'd be great to get, do you have other plans for the sediment cores that are on the side of the sub?
We do not. So
If possible, that's a living clam bed. If you could get, if we could do some close-up imagery of the clam bed and then put a few sediment cores in there,
Sure.
that would be wonderful.
Sure thing. You want us to sit down and just take close-ups on everything and anything and everything here?
Yeah, that'd be wonderful.
All right.
Andrew has perfect timing.
Hmm?
He came in
Yep.
right as I got CP.
Oh, I've been watching and listening downstairs. I've been working on dive plans.
Okay then, intentional, perfect timing.
My luck's not that good that I just wander in right when we found a seat.
We might settle in over the top of you or a little further. You want me to back up, or are we okay? I think we have about probably 20 meters of latency right now. Okay. So we should maybe bring it back a step. Not necessarily. We can also just turn Argus around. Okay. If you want, we can do a 10 meter back the other way so that we get out a little bit of the a little bit of this way.
So Jenny,
You want to bump out?
Andrew,
cliff?
can use
Sorry.
push cores if needed.
Actually, I think we're all right, honestly. Yeah. Would you mind getting down on the stick lock there? Yeah. Thank you. All right, Andrew, so probably this clam bed here right in front.
Yeah, can you zoom in a little bit? I want to see if there's any clams with their siphons out.
Sure thing, go ahead on NL. How tight do you want it there, Andrew?
Actually, I can see right there. If you can zoom up just a little bit.
Sure. Let me know when.
And just, that's perfect right there.
Okay.
So this is really beautiful because it's not only a bunch of large living clams, these are vesicomyids. You can also see all those little worm tubes, and so those are amphoretid polychaetes, which are also really indicative of seep habitats. So this is a really exciting discovery. For sampling, I'd love to get some cores sort of right here and here next to each other. Other great options are there and there. If we can put about...
we're not
Are you
seeing
pointing? Are you telestrating?
Oh, I'm telestrating.
We didn't see it.
It's
We didn't see it.
It's not showing up on your screen.
Where's our telestrator?
Give me a second, I'll work on that.
Oh no, you're good. I'm just checking.
I don't know where our pokey stick went. That was the old telestrator. So where Andrew is pointing is in the top left quadrant of this current image, where there's kind of a gap in the clams.
So just to the left of the lasers?
Yeah.
Okay.
And that little spot without all the big clams in it?
All right, and that is a tube core?
Yes, please.
Just left of the lasers, roger. Guys clear to come full wide? Andrew, you got all your shots?
Yeah.
And Andrew, could you give me a little telly so I could see if I found the right source?
Oh, when you get a sec, can you come full
Excellent.
wide, please?
Thank you. Yes, we'll do.
Thank you.
Nice to see the seep just right around the corner from the